Drosophila larvae bearing a neuron-specific CD8 GFP fusion reporter transgene are used to isolate DA neurons. The larvae are dissected by inverting their cuticle inside out, exposing the peripheral nervous system. The inverted larvae are subjected to enzymatic dissociation, followed by mechanical dissociation via vortexing, trituration, and downsing. Cells are then filtered to obtain a single-cell suspension containing primarily epithelia, muscle, and neurons. Antibody-coated magnetic beads are added to the cell suspension, where they bind neurons expressing the CD8-tagged GFP fusion protein. Finally, the magnetic bead-coated neurons are isolated by placing the solution in a strong magnetic field, yielding highly purified neuron populations.
Hi, I'm Dan Cox from the Department of Molecular and Microbiology and the Krasnow Institute for Advanced Study at George Mason University.
Hi, I'm Eswar Iyer from the laboratory of Dr. Dan Cox.
Today we will show you a procedure for the isolation and purification of Drosophila peripheral neurons by magnetic bead sorting.
We use this procedure in our laboratory to study the molecular mechanisms regulating class-specific dendrite morphogenesis. So let's get started.
To begin this procedure, pick 30 to 50 age-matched third instar larvae and place them in a 1.6 milliliter microfuge tube containing 1 to 1.2 milliliters of 1x PBS. Close the tube and vortex it at the maximum setting 3 times for 1 second each. Using a fire-polished Pasteur pipette, discard the supernatant completely, add 1 milliliter of PBS, and repeat the water.